The interplay between nicotinamide mononucleotide, NMN, autophagy, and the gut microbiome offers a compelling and emerging view of how cellular renewal mechanisms intersect with microbial ecosystems to shape metabolic health during aging. As individuals grow older, the gut undergoes significant changes. Barrier function weakens, microbial diversity declines, and the balance of pro- and anti-inflammatory microbes shifts toward a dysbiotic state. These disruptions contribute to systemic inflammation, insulin resistance, and obesity. Autophagy, a cellular self-repair and recycling process, is deeply involved in maintaining intestinal barrier integrity, regulating immune tolerance, and shaping microbial composition. NMN, by replenishing NAD+ and enhancing autophagy in intestinal cells and immune networks, may restore gut microbiota homeostasis and offer protection against metabolic deterioration in elderly individuals. Aging is known to impair autophagy across tissues, and the gut is no exception. In intestinal epithelial cells, autophagy ensures turnover of tight junction proteins, mitochondrial quality, and defense against oxidative stress. It also plays a critical role in controlling the secretion of antimicrobial peptides, mucins, and immunoglobulin A, IgA, all of which maintain a stable and diverse microbiota. With diminished autophagy, the intestinal lining becomes more permeable, leaky gut, allowing bacterial metabolites such as lipopolysaccharide, LPS, to enter the bloodstream and trigger systemic inflammation, a major contributor to age-related obesity. NMN, by increasing NAD+ and activating SIRT1 and AMPK pathways, re-engages autophagic flux in gut epithelial cells and associated immune cells, helping to restore barrier function and reduce inflammation at its source. Animal studies have shown that NMN supplementation reverses age-related thinning of the intestinal mucosa, improves gut permeability, and reduces LPS levels in circulation. These improvements correspond with elevated expression of autophagy-related genes, e.g., LC3, Beclin-1, and tighter regulation of mitochondrial dynamics in enterocytes. This restoration of cellular housekeeping leads to a healthier gut environment, where the host can better regulate microbial populations and suppress the overgrowth of pathogenic species, often seen in aging and obesity. Additionally, by supporting Paneth and goblet cell function, cells responsible for secreting antimicrobial agents and mucins, NMN-induced autophagy contributes to the chemical defense of the gut lining, further stabilizing microbial ecology. Interestingly, NMN's influence on the microbiome may also extend to changes in microbial composition itself. Preliminary data suggests that NMN supplementation can increase the abundance of short-chain fatty acid, SCFA, producing bacteria such as Akkermansia mucinifila and Faecalibacterium prosnitzi, which are associated with improved gut barrier function, anti-inflammatory effects, and enhanced metabolic regulation. These SCFAs, especially butyrate, have reciprocal effects on the host, promoting autophagy, tightening junctions in the gut lining, and supporting energy homeostasis. In this way, a positive feedback loop emerges. NMN enhances autophagy, which improves gut health and microbial composition, and a healthier microbiome in turn supports host metabolism and further stimulates autophagy. In the context of obesity and metabolic syndrome in older adults, this NMN, autophagy, microbiome axis could represent a transformative mechanism. A dysbiotic gut contributes to low-grade inflammation, insulin resistance, and abnormal fat storage, even in individuals who do not consume excessive calories. By restoring microbial diversity and function, NMN may help reduce intestinal and systemic inflammation, lower endotoxemia, and increase metabolic flexibility. The observed improvements in hepatic and adipose tissue function seen in NMN-supplemented aged models may, in part, be mediated through this gut-centered pathway. Given that the gut, liver, and gut-brain axes are deeply interconnected, NMN's influence on the microbiome may extend beyond digestion and immunity to affect broader systems, like glucose regulation and appetite control. While human data are still emerging, the available evidence strongly supports the plausibility of NMN as a modulator of gut health via autophagy. Early trials in elderly individuals have shown improvements in insulin sensitivity and inflammatory markers that could plausibly be mediated by better gut barrier function and microbial balance. If future research confirms these links more directly, through metagenomic and metabolomic profiling before and after NMN use, It could open up new therapeutic strategies focused on microbiota rejuvenation in aging populations. In summary, NMN's effects on metabolic aging are not confined to energy metabolism or organ-specific rejuvenation. They also appear to involve deep, regulatory communication with the gut microbiome, mediated in large part by autophagy. This triad, NMN, autophagy, and the microbiota, constitutes a feedback-rich system that may protect elderly individuals from the inflammatory, metabolic, and immune disruptions that drive age-related obesity. By restoring cellular renewal in the gut and rebalancing microbial ecosystems, NMN may offer not only longer life but healthier years, driven by a cleaner interface between the host and its microbial partners.